Alright, hi everybody. So I recently had a student and also a friend of mine who's taking AP Biology ask me how I went about approaching the FRQ problems on the AP exam. So I thought I'd make this quick video, well, I don't know how quick, but I thought I'd make a video showing how I tend to personally approach the FRQs on the AP Bio exam. And I hope this is helpful. So to those that asked, this is for you if you're watching. So the FRQ was about meiosis, cell division. So I thought before I do the FRQ, I'll just give a little brief overview of meiosis just so we're all on the same page and we can approach the matter and the material together. So meiosis has to deal with genetics. So in the human body, your genes are made of these long strands of DNA. Remember, DNA is a double helix, and it contains the genetic code for storage in your body. Now, DNA, it looks like this. It's a long strand, but in the human body, it's very inefficient to store DNA as this big, long strand. So what happens is your body compresses them into 46 chromosomes. Let me just check to make sure it's still recording. All right. Looks like we're good. So your body condenses the DNA into 46 chromosomes. So it takes this long strand of genetic material and it packs them into these 46 things that look like this. It's a chromosome, like that. Alright, so this is how the DNA is stored. All this matter you see here is made up, wound up DNA, is made of DNA. So just a little basic anatomy of the chromosome. So let's just talk about the different parts we have here. We have this part right here in the middle. See this red thing? I'm going to color it here. This is very important. This is called the centromere. The number of centromeres determines the number of chromosomes. So when we count chromosomes, we count it by the number of centromeres. And the reason being is because sometimes in the body, chromosomes look like this. Sometimes they might look like this. And it depends on which stage of the cell cycle you catch the cell during when you look at the chromosome. So then the way scientists define it is this is one chromosome because it has one centromere, the center part. This is also one chromosome because it also has one centromere. Even though this one looks like it has more parts, it's still one chromosome because we count chromosomes by the number of centromeres. So then this would count as one chromosome, and this would also be one chromosome, because they each have one centromere. So, other parts of the chromosome, if you look at, I'm going to try to circle it, this part of the chromosome, this one half here, this is called a chromatid. Sometimes you might hear them be called sister chromatids. And likewise, this on the other side is another chromatid. This is also one chromatid here. But then what happens is, if you look closely, this one has more chromatids because each one of these halves is a chromatid. So if we look here, this one has two chromatids. Let me just write it in blue so it's more simplistic. So this is two chromatids because it has two halves. This is one chromatid because there's only one half. It only looks like a stick. But then both of these, since we count chromosomes by the number of centromeres, since each one only has one centromere, this here is one chromosome, and this here is one chromosome. So just an important distinction, how they're both one chromosome, but this one has two chromatids, this one only has one chromatid. Alright, so just some basic anatomy right there. That's how chromosomes are organized. And this is what really confused me when it came to mitosis, meiosis. I didn't understand, is this a chromosome, is this a chromosome? It just depends when you see it in the cell cycle. So another distinction I'll give you is when does a chromosome look like this and when does it look like this? It looks like this before the S phase. and it looks like this after the S phase. And if we remember from the cell cycle, the S phase is when the DNA replicates. So this is what it looks like before the DNA replicates. This is what it looks like after the DNA replicates. And it makes sense, right? Because if the DNA is replicating and doubling, it makes sense that we're going to get twice as many chromatids, but still only have one chromosome, because we only have one centromere. All right? So let me just think if there's anything I'm forgetting. All right, so now we can go to the cell cycle. Mitosis, you guys talked about already. Now let's look at meiosis. This is how sex cells are generated. And fun fact, sex cells are sperm cells and egg cells, but they're actually different. A sperm cell replicates differently than an egg cell. But that's something you'll cover in genetics or anatomy physiology if you do ever take that in college. All right, so we're looking at this. This is a cell. All right. Each one of these little lines that you see, these toothpicks, is a chromosome. So how do we know that? Because if we look here, I don't know if you can see on the camera, you see each one has a little dot at the center. That's the centromere. So there's a centromere and there's a centromere. So since each one of these little twigs has one centromere, each one is one chromosome, all right? And if you count all of them, you'll see that we have 46. All right? Now, this is what our genome looks like before the S phase, which means it's before the DNA replicates. All right? So if you notice here, we have 46 chromosomes. If you notice, some of them are in blue and some of them are in red. The blue ones are the ones that come from your father. You got the blue ones from your father, and now the red ones are the ones that you got from your mother. And then if you count each one, and hopefully I didn't add or subtract an extra chromosome by accident, if you count each one, you're going to notice that you have 23 blue chromosomes, and you have 23 red chromosomes. So 23 blue chromosomes came from your dad, you got them from your dad, and 23 red chromosomes came from your mom. All right? So this is what it looks like before we replicate, before the S phase. Then we go on, and we go through the S phase. Now it looks like this. So this is after the S phase. This is after the DNA replicated. So after DNA replication, I'll just abbreviate that. So if you notice, before we replicated, it just looked like little toothpicks. Now it looks like little X's because the DNA copied itself during the S phase. But if we look carefully, you see how each one still has only one dot at the center? Just like before, each toothpick only had one dot, now each X only has one dot. That means that each one still only has one centromere. So remember, we count chromosomes by the number of centromeres. So this is one, this is one, this is one, this is one. So if we count all of them up, we're going to notice we still have 46 chromosomes, even after we replicated. And once again, when we count the blue ones, we see we have 23 blue chromosomes from our dad and 23 red chromosomes from our mom. So this is what it looks like after we replicate, all right? And that's after the S phase. Now we're going to keep going. When we're going through meiosis, we're going to go through something called prophase, prophase 1. I know it's a little hard to see on the screen. Oh man, I gotta move this. Let me just refocus it. Alright, so here we are at prophase 1. During prophase 1, what happens is you still have 46 chromosomes, because if you count the number of centromeres, we still have 46. But what happens is the blue chromosomes pair up with their corresponding red chromosomes. Alright? So if we look here, you see a blue chromosome and a red chromosome, a blue chromosome and a red chromosome. When we're talking about, let's say just focus on this one here. We have a blue one and a red one. This is called a homologous pair. Or sometimes you'll hear them called as homologous chromosomes. Homologous means that you got one from your dad and one from your mom. So both of these are similar in genes, but it just so happens that one came from dad and one came from mom. So during prophase 1, the homologous chromosomes pair up, which means that a chromosome from your dad pairs up with a chromosome from your mom. So the way I like to think about it is prophase 1, and right up here, this is the hookup phase. The hookup phase. I don't know if you can see that pen writing. So the homologous chromosomes are hooking up. Your dad's chromosomes are pairing up with your mom's chromosomes, and they're doing something called crossing over. I'll write that here too. It's crossing over. And what happens is they're holding hands, because if you see how the red chromosomes are holding hands with the blue chromosomes. They're holding hands and exchanging some information. So they're swapping some of their genetic code. Alright? So when they exchange information, this leads to something called recombination. Recombination is a fancy word for saying we're shuffling. Like shuffling a deck of cards. We're shuffling genes. So some of your dad's genes are mixing a little with some of your mom's genes. And basically the blue and the red chromosomes are hooking up and they're swapping. They're exchanging some genes. That's what leads to genetic variation. That's why kids are never like their parents when they come out, because some of the genes have been exchanged. Alright? So that's prophase 1. There's crossing over. So when you think of meiosis, prophase 1, think of crossing over. Now we're going to keep going. We're going to go to metaphase 1. So we just finished prophase 1. The chromosomes are hooking up. They're on their date. Each one's got a partner. During metaphase 1, they're still going to hold on to their partner, but they're going to line up along the middle line. Alright? So during metaphase 1, you notice how the blue chromosome, each blue still has a red. They're still lined up, they're still holding hands, they're still attached to each other, but now they're lining up on this imaginary midline along the inside of the cell. Alright? So what happens is, this is the neat part with cells. When the chromosomes are lining up, there's a choice to be made. You notice how they're lining up on the line. The cell has to pick, do I put the blue chromosome on the left or do I put it on the right? So do I put dad's chromosome on the left side of the line or do I put it on the right side? And what happens is the cell just makes a random choice. It can put this blue chromosome on the left and this one on the right and this one on the left and this one on the right. And that's the reason why sometimes you inherit genes more from one parent than the other. So some kids get more genes from mom and some kids get more genes from dad. And this is why, because during metaphase 1, when the homologous chromosomes are lining up on the midline, there's a random choice. Do I put mom's chromosome on the left or on the right? Does the blue go on left or right? And it really doesn't matter. Alright? Something else that happens during metaphase 1 is these things right here start to form. These are called centrioles, or sometimes you hear them called centrosomes. There's a difference. I can't quite remember what it is. But they're here. They start to produce spindle fibers. And the spindle fibers from each side are going to grab on to the chromosomes. So I'm going to draw, I don't know if I'll have time to draw them all. each spindle is going to grab onto the chromosome on its side. And it's going to grab onto the middle part, the centromere of the chromosome. There's a special structure on the centromere. It's called the kinetochore, and that's what it's grabbing onto. So I'm just going to draw them in really quickly. So the spindle fibers are grabbing on. That's metaphase 1. Now we go to anaphase 1. So the spindle fibers, let me just let it focus, are actually starting, this is anaphase 1, to pull the chromosomes apart. So let's just recall from metaphase 1, the blue chromosomes and red chromosomes were still holding hands. By the time we get to anaphase 1, they're pulled apart. We pulled them apart. But if you look closely, I don't know if you can see it, you see how even after we pull them apart, this blue chromosome still has a little bit of red attached to it, and this red chromosome still has a little bit of blue attached to it. So they ripped off of each other's body part of each other's body, and still carrying it with them even as they get pulled to the other side of the cell. So that's how genetic recombination happens, which is a fancy way of saying that's how genes are shuffled around, like shuffling a deck of cards. Even after the chromosomes, the homologous chromosomes get separated, they still have a little bitty part of the other chromosome that they hooked up with. And that's how genes change over time. Alright, so now these spindle fibers, I'll just write it here. These are called spindles. Spindle fibers are pulling the chromosomes apart to each side of the cell. And then once they reach the other side of the cell, the membrane starts to split, and we get telophase 1. So telophase 1 looks like that. And we see how, if we look closely and count chromosomes, let's just count 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. We have 23 chromosomes in this cell and 23 chromosomes in this cell. Because remember, we count chromosomes by the number of centromeres. And each one of these little X's only has one centromere. So it's 23 here and 23 here. Alright? So remember how at the very beginning, at prophase 1, we started with 46 chromosomes in this one cell. But then now, after one round of division, we only have 23 in each cell, alright? And now each one of these cells gets ready to divide, and we go through prophase 2. Now prophase 2 is not very exciting. Each cell basically starts to condense again, get ready to divide, but there's no hookup phase in prophase 2. So prophase 2, there's no hookups, which means there's no crossing over. So the genes don't shuffle anymore. After prophase 2, we're going to go to metaphase 2. So we still have the two cells, but now notice how the chromosomes are lining up single file. Alright, that's very important. The chromosomes line up single file this time. Now, I'm just going to bring back the picture of metaphase 1 so we can remember what it looked like. You see how in metaphase 1, the cells hooked up? They got in partners and then they lined up. In metaphase 2, they're just lining up. There's no partnering or anything. They're just lining straight up. So that's metaphase 2. Alright? And once again, the spindles are going to form, which unfortunately means I have to draw all of them. I'm just going to draw some of them. For the purposes of this, just assume that I drew all of them in. If we take a closer look, we notice we still have blue and we still have red. We still have some from Dad and we have some from Mom. But if we count how many chromosomes in each side, we still have 23. We still have 23. Alright? It's still 23. Finally, we go to anaphase. Anaphase 2. And once again, the spindles are pulling the chromosomes apart. But if you notice here, the chromosomes get pulled apart, but the centromeres also get pulled apart. So now each one of these dots is a centromere, so each one of these little twigs is a chromosome, which means that our chromosomes, in theory, are doubling here because we're splitting apart the centromere. So I'm just going to draw it closer so you know what I'm talking about. This is what it looked like in metaphase 2. And during anaphase 2, what we're doing is we're doing this. You see how we're ripping apart this little centromere in the middle and it's splitting in half? So now this is 2 chromosomes now because there's 2 centromeres. Which means that by the time we get to telophase 2, so each one of these split in half gives us this. Each one of these twigs still has one centromere in the middle now. It's 1 centromere, and we count chromosomes by centromeres, so we count how many centromeres we have here, and we get 23. So that means this cell has 23 chromosomes. This one also has 23. This one has 23, and this has 23. All right? So that's meiosis. And that's the process of splitting up chromosomes. So now a common thing people ask is, now that we went over the basics, how do you study this? How do you keep straight? When do you have 23 chromosomes? When do you have, 46 chromosomes? Here's how I like to study. I like to draw it as a flowchart. So we start with, we started with prophase 1. Then we went to metaphase 1, anaphase 1, telophase 1, then prophase 2, anaphase, and metaphase 2, anaphase 2, telophase 2. Alright. So now let's keep track of how many chromosomes we had at each one. Prophase 1, we started with 46. I'm just going to look back at our pictures. Prophase 1, right, we have 46. Metaphase 1, we still have 46. Anaphase 1, if we look here, it's coming apart, but if we count them, we still have around 46, but it's splitting up. And then by the time we get to telophase, we have 23. Alright, so from anaphase 1 to telophase 1, that's when we're cutting the number in half. And then we go to prophase 2, each cell has 23. Then we go to metaphase 2. Let me just look for where metaphase 2 is. Metaphase 2, each cell has 23. And then we go to, we're going to go to anaphase 2 and telophase 2. Anaphase 2 has 23, and telophase 2 has 23. So, this step right here, anaphase 1 to telophase 1, that's the key step where you're cutting the number of chromosomes in half from 46 to 23. So this is how I study for it. I just make a little flowchart like this, abbreviated, so you can remember, alright, this is what it looks like, these are the numbers, and then from here to here, that's about when it changes. Let's see. Is there anything that we... Oh, alright. Alright, so another thing people often ask is, how do you study the vocab? How do you make sure you remember all the information? The best piece of advice I can give you, and the way I did it in college, was I try to make a list of all the words, all the vocab words. So let's try to do that. Go through meiosis in your head again and try to make a list of all the words that we covered. So what did we talk about? We talked about DNA. We talked about chromosomes. We talked about chromatids. We talked about centromeres. We talked about the S phase, right? We talked about prophase 1, metaphase 1, talked about anaphase 1, we talked about telophase 1, and then we also talked about prophase 2, and metaphase 2, anaphase 2, telophase 2. And then we also talked about some other parts too, such as centrosomes or centrioles. We talked about spindles. We talked about the membrane. Oh, we talked about homologous chromosomes. That's a big one. Alright, those are the big ones I can come off the top of my head. So the way that I study for a test is I make a list of all the words. Sometimes it's like many pages. And what happens is when you're studying the week before, you should be able to go through each of these words and define what it is. So, like DNA, define it in your head. That's the genetic code in our body, right? Chromosomes, we have 46 of them. It's what the DNA is packed into when it's stored in the body. Chromatids, it's the half of the chromosomes. Centromere, it's the little centerpiece of the chromosomes that we use to count them by. So go through the list, try to define them in your head, and if you can't define one, go back and rewatch the part of the video that we talked about earlier. And this is how you study. As the list gets longer, you go through each of the words again every day. So by the time you sit for the test, there's no vocab word that should be able to surprise you because you've seen them all and you've organized it into one place. All right? So that's my biggest tip with studying. And probably the best thing you can do for something like this is just to draw it out. Make your own drawings. And teach a friend. Teaching is the best way to learn because it shows that you're understanding it. Let me just make sure the recording's still good. All right. The last thing I wanted to do is I wanted to, I forgot a word. That's an important word. Recombination. And there's probably other words that I just can't think of right now, but you can find them as you go through the video. The other thing I want to go through is one of the FRQs for AP Bio. Now, FRQs I struggled with. I really did struggle with them when I took AP Bio and AP Chem and AP Physics too. But these are important nonetheless. So, unfortunately, we have to learn how to do them. I'm just going to show you my thought process when I'm going through one of these and how I approach something like this. So we get this on the exam. We flip the page. We see this. You're probably going to say a lot of bad words in the moment because, oh my God, what the hell? And, alright. So, initial shock, initial internal crying is over. Now we're going to approach it, alright? So, let me just make sure. Alright, I'm going to show you what goes through my head when I read one of these. So we're going to play a game called, what I do is I play a game called Caveman, which is translating all this junk into plain and simple English. So during meiosis, double-stranded breaks occur in chromatids. Okay, what's a chromatid? Remember, it's that one half of the chromosome that we talked about earlier, alright? Double-stranded breaks. The breaks are either repaired by the exchange of genetic material between sister chromatids, which is crossing over, or they are repaired without crossing over. So, all this junk up here, I'm going to try to summarize. What's the main point? We have double-stranded breaks, which is when there's a break in the chromosome like this. I'm just going to abbreviate DSB. Double-stranded breaks can lead to recombination. Remember, recombination means shuffling of genes. It can lead to genetic exchange and crossing over. So recombination is another way of saying crossing over. It's the process crossing over is what causes recombination. So double-stranded breaks can cause recombination and crossing over, or nothing at all. So one or the other. So because it says it can either cross over or it can just be repaired without anything happening at all. All right. So now we know double-stranded breaks, we can cross over or not. All right, that's the gist. Plant breeders are developing new varieties of corn and want to see if there's a correlation between the number of double-stranded breaks and the number of crossovers. So the question that they're asking is, does more double-stranded breaks cause more crossing over? Is there a relationship between the two? I don't know. But let's keep reading and find out. Using specialized staining and microscopy, don't worry about that. That's garbage. Scientists counted the number of double-stranded breaks. This is so important. They counted the number of breaks and the number of crossing over. And the same number of... don't worry about that. Alright, so they give us the table. And I hate reading tables. But we can approach this together. We can do this. So the first thing you do is you look at the column headers. This is the number of double-stranded breaks, and this is the number of crossovers. So I'm just going to scan through. I'm going to see, is there any relationship I can just see based on just scanning through briefly. So I look here at 710, 650. It looks like this is going down. 19, 18, 17, and it looks like this is going down too. So immediately off the bat, it looks like as the number of double-stranded breaks decreases, as the number of breaks decreases, it looks like the number of crossovers decreases. Alright? I can just see that based on reading the table. I don't know what it means at this point, but just keep that in the back of my head as I go forward. Alright, next page. We're getting somewhere. Alright. Double-stranded breaks occur along the DNA backbone. Describe the process in which the breaks occur. Alright. This is kind of hard to explain without having taken organic chemistry and biochemistry. So unfortunately for part A, I'll answer it down here. Unfortunately, that's just something we have to memorize. And I'm looking at the answer key right now. And it says that the process in which it occurs, you can either say hydrolysis between sugars, phosphates, and nucleotides, or between the covalent bonds, or the covalent bonds between them are broken. One or the other, but unfortunately this is one of the questions you just have to memorize. So I'm going to say, one thing I can say is enzyme hydrolysis between the phosphates, between the sugar, between nucleotides. Or another thing I can say is the covalent bonds between sugars, phosphates, nucleotides are broken. That's what the answer key says. So we just have to know that off the top of our head. Alright, the next thing is using the template space provided, construct a graph that represents the data and allows us to examine a correlation between the two variables. Alright, so this is basically, it's a lot of words, but the big takeaway is it's telling us to graph. So let's graph it. So part B, we have to graph it. I would be honest, I don't like graphs either, but we don't have a choice in this case. So I'm just gonna rough sketch it. Let's bring back our table here. I'm gonna put double-stranded breaks on the x-axis, and we're gonna put crossovers on the y-axis. And I just do that just from intuition. Double-stranded breaks, based on the way they word the problem, it sounds like the independent variable because it's affecting the number of crossovers. So then I look here, I'm gonna put like a scale, 200, 300, 400, etc. 10, 12, etc. Now, I'm going to go, let's graph the lowest one first, 325, so I'm going to go over 325, and it looks like we have, when there's 325 double-stranded breaks along the x-axis, it looks like we have 11 crossovers. So I'm going to go 325, I'm going to go up to 11. And it says it's plus or minus 1.5, which means I'm going to go 1. Oh, that's way too big. Oops. I'm going to go 1.5 up, and I'm going to go 1.5 down. And I'm going to keep doing that all the way for all of these values. And it's going to look like this. That's just the answer key. And then the second part of the question asked me, where is it? Based on the data, determine whether corn strands 1, 2, and 3 differ in the average number of crossovers. So, we look here, strands 1, 2, and 3. I'm going to look on the graph that we made, and it looks like they're talking about this. Do they differ in the average number of crossovers? So, the number of crossovers is going to be on the y-axis. And it doesn't really look like they differ much. So it's not a big difference, but the answer is it's not as statistically, I can never say that word, it's not statistically significant. Alright? It's not statistically different. One thing we can say is that strain 1 is higher. Notice how strain 1 is the highest number of crossovers based on the table compared to all the other strands because it's the highest up the graph. But we can't really say much else about it. So I guess what we can say here is not statistically significant between strands 1, 2, and 3. Of course, on the FRQ, you would explain this in a sentence or two, but that's what I would say. Alright, part 3. Based on the data, describe the relationship between the average number of double-stranded breaks and the average number of crossovers. So, this is asking, what's the takeaway? Why do we care? Alright, what's the takeaway? Well, let's go back to the data here. The data table, or the graph. So I'm going to put them side by side, and we're going to notice here, as the number of double-stranded breaks decreases, the number of crossovers decreases. On the flip side, if the number of double-stranded breaks increases, the number of crossovers increases. And we can see that on the graph here. As we get more double-stranded breaks, we get more crossovers. Alright? So, that means as x increases, y increases. So that is what we call a positive relationship. So I'm going to write that down here for my answer. There is a positive relation. There is a positive relationship. or sometimes it's called a direct correlation, between the number of double-stranded breaks and crossovers. Which means as the double-stranded breaks numbers increases, the number of crossovers increases. Alright? So pretty simple. So that's described. And we described it. We talked about what it was. Now, number 4. Oh god. Letter D. This is going to be a pain in the ass. So, it says, crossing over creates physical connections that are required for proper separation, which means if we don't cross over properly, there might be a problem with division. So a diploid cell with 4, oh boy. As soon as I read this, I'm saying this is going to be a scenario question. Alright, but we can do it. Crossing over, so a diploid cell with 4 pairs of homologous chromosomes undergoes meiosis to produce 4 haploid chromosomes. To 4 haploid cells. Crossing over only occurs between 3 pairs. Predict the number of chromosomes in each one. Alright. So, this was a hard problem. And this is something they would ask in college, for sure. I'm just going to draw it out. So, 4 pairs of homologous chromosomes. So, let's remember, homologous chromosome means that it's one from each parent. So do you remember in our old drawing there was a blue chromosome and a red chromosome? So 4 pairs of homologous chromosomes means you have 4 of these. Alright? Now, I just color-coded it so you can see what they're talking about. So each one of these is a pair, so it's 4 pairs and 8 chromosomes total, because there's 2 chromosomes in each homologous pair. Alright, so each one of these, this is a homo pair. And it says that only crossing over happens in only 3 of the pairs. So, only 3 of them cross over. So, based on the reading here, it says, crossing over is required for proper separation. So, if crossing over is required to separate, not crossing over means it's not going to separate, right? Just by logic. So let's say this pair here doesn't cross over. So there's an error in this pair. Alright? Now let's say it's going to divide. Let's say the cell divides. This is going to be hard to visualize, but let's see if we can do it. We're going to go through metaphase 1. And anaphase 1. So, let me just remind you guys what that looks like. Metaphase 1, it looks like this. Anaphase 1 looks like this. The chromosomes, the whole thing is getting separated. So these 3 pairs crossed over, so they're going to separate. But since this one didn't cross over, it's not going to be able to separate. So that means when the spindle fibers come, and they pull each one of these apart, this one's not really going to get pulled apart. This one can't get pulled apart because there was an error. So it means the whole thing is going to get dragged this way, the whole thing is going to get pulled this way, or the whole thing is going to get pulled this way. So let's say it gets pulled this way. That means when we separate them into their own cell, one of the cells is going to look like this. One of the cells is going to look like that. So let's count how many there are. 1, 2, 3, 4, 5. This cell has 5. This one has 1, 2, 3. That's 3. And why does it have 5? Because these chromosomes couldn't separate. Because, as the problem stated, crossing over only occurred in 3 pairs. So this one didn't cross over, therefore it couldn't separate. That, in biology, is what we call non-disjunction. Non-disjunction. That means failure to separate. Alright? Alright, so now let's say we went through one round of division, a metaphase 1 and anaphase 1. Now this is going to go through meiosis 2. This is going to split. Again, this is going to go through metaphase 2 and anaphase 2. Let me see if I can find those pictures. Anaphase 2. So each one of these little chromosomes is going to get separated. So now it's going to look like 2, 3, 4. This. So this is going to be a cell, this is going to be a cell, this is going to be a cell, and this is going to be a cell. So we'll count how many chromosomes. This one has 5, because remember we count by centromeres. This one has 5, this one has 3, and this one has 3. And that is your final answer. So for part D, let's say we were going to answer it on the exam. I'm going to write my answer for D. We're going to say 2 cells have 3 chromosomes, and the other 2 cells, because we produce 4 cells, have 5 chromosomes. Alright? And the reason being is because one pair one homologous pair, it's this one we're talking about, did not separate in meiosis 1. But it did separate in meiosis 2. Now, why does it separate in meiosis 2 but not meiosis 1? That's a whole other story. Ask me again in 3 years when you take genetics in college. But there's a reason, but we're not going to get into it. It doesn't separate in meiosis 1, but meiosis 2 happened normally. Alright? Now, of course, never bullet point your answers in the FRQ like this. You would write them in short sentences. But I don't want to take up too much of your time writing full sentences, so I'm going to abbreviate here. So, that's part D. The last part here, let's see, we predicted, we did that, we justified. The last part is explain how plant breeders can use the information in Table 1 to develop new varieties of corn. Alright, so this is one of those application problems, and we really have to think outside the box to answer these. So, let's look here. So, do we remember what crossing over does? You can think about it yourself. I'm going to write it here. Crossing over can see the handwriting. Crossing over increases genetic diversity. Alright? More genetic diversity means that there's more types of corn. Because that means that the different types of corn will have different genes, and different genes will lead to new features. Maybe they're going to have red kernels, or blue kernels, or yellow kernels, or maybe they'll be bigger and juicier, or whatever. That all comes back to genetic diversity. So crossing over increases genetic diversity. We see here from Table 1 that as the number of double-stranded breaks increases, the number of crossovers increases. So, what we can do is we can say, since more double-stranded breaks leads to more crossovers, what plant breeders can start to do is they can breed strains that have more double-stranded breaks. Alright, or they could breed plants that have more crossovers. They could start to carry more plants with more crossovers and more double-stranded breaks, because the more crossovers you get, the more genetic diversity and the more types of corn you get. Somehow, if you can find a way to increase double-stranded breaks too, that will also lead to more crossing over and more genetic diversity. So, let me just answer that. This is also going to be my answer for Part D. What I'm going to say is, I have to word it well for the AP exam. Breed strains with higher rates of crossing over or double-stranded breaks, because that will increase genetic diversity, which leads to more varying strains of corn. Alright? Leads to more varying strains of corn. So, just by looking at this, looks like strain 1 has the most number of crossovers, right? So that's the type of corn. If I'm a plant breeder, I might want to carry this, or a corn grower, I might want to carry this kind of corn because there's a lot more variation involved in it. Alright, so that is the gist of our FRQ, our little FRQ lesson, and our little study lesson, and Meiosis lesson. I hope that that was helpful to you guys. Let me know if you ever need anything else. Otherwise, good luck. Have a great, happy start to the new year. We made it to 2024. Have a good time, and best of luck to everyone. Thanks. Thanks for listening.